Hello. Good morning to all of you. Dear students, warm welcome in the cold morning. I welcome our Joint Director Research and Head Division of Genetics, Dr. A.K. Singh. On the inaugural day, you listened to the glimpse of his teaching ability. He is a teacher for excellence. I'll briefly introduce you to him, although most of you, those who are from IRI and other places, those who are doing genetics will be knowing him very well. You might have read his books also. So Dr. A.K. Singh was born in village Barahat, Ghazipur, India in July 1962. He has done his undergraduation and master graduation in agriculture from Banaras Hindu University. He completed his masters in agriculture in 1985. After that, he did his PhD from Indian Agriculture Research Institute. Thereafter, he served in different institutes. Initially, he served in Tata Energy Research Institute, New Delhi. Later, he served in the Bioseed Genetics India Private Limited in the industry. And then he joined in IRI in 1994. Since then, he is working in IRI at various capacities. He served as scientist, senior scientist, principal scientist, and professor of the Division of Genetics. And later in 2014, he is joined as the head Division of Genetics. And currently, he is also working as Joint Director Research of the Indian Agriculture Research Institute. His main research areas are rice breeding, specifically molecular breeding of rice. He has 29 years of research experience, including 24 years of teaching experience at IRI. His research has made a lot of significant contribution to the country as well as the welfare of the farmers. The first mass-derived variety of rice, that is improved Pusa Basmati 1, wherein the molecular markers have been used and the variety was improved for disease resistance. It was released by Dr. A.K. Singh. And later, the Basmati rice variety, Pusa Basmati 1121, which is earning more than 30,000 crores foreign exchange every year, is released by him. And then he has also improved the Pusa Basmati 1121, and the improved varieties are now picking up. They are occupying the more than 95% area of the Basmati grown area in the country. In addition to his research contribution, he is a teacher par excellence, one of the best teachers IRI has ever had. So with all his significant research contribution and teaching achievement has been duly recognized by the system. He has received IRI Best Teacher Award in 2003, and he has received BP Paul Memorial Award, and he has received Borlaug Award in 2012. And the highest award for teaching in the ICR system, that is ICR Bharat Ratna Dr. C. Subramaniam Award for Best Teachers in 2013. He has also received the ICR's highest award, that is ICR Rafi Ahmed Kidwai Award for Outstanding Research in 2013. He is a fellow of Indian National Academy of Science, that is FNA, one of the highest scientific academies of the India, very difficult to get FNA. And he is also fellow of the National Academy of Agriculture Sciences. And I have a very long biodata of him. Since he told to make it brief, I am stopping it here. Sir, with this brief introduction, I welcome you to deliver your lecture. Thank you very much, sir, for kindly accepting and coming to deliver the lecture. Thank you, sir.
Thank you. Thank you, Dr. Viswanathan, for generous introduction. And very good morning to each one of you. Friends, what I'm going to do is to talk to you about some of the research activities that we have in Basmati rice breeding program, integrating molecular markers, but then finally correlating it how the new tools and techniques like genome editing can be helpful in future. And the preliminary background that I will prepare will really help you to understand what Dr. Reddy is going to present. What I am going to talk about is more of a concept-like thing. What Dr. Reddy is going to present before you is actually what is that he is doing for rice improvement using genome editing tools. But I shall be presenting primarily a breeder's perspective and certain basic principles that you need to understand when you are integrating the genome editing technology in the breeding program. What are the pros, cons, consequences that you will be facing and that you should be aware of. Okay. Now, it has been very well pointed out in the first inaugural day by Dr. N.K. Singh, and subsequently in his lecture I watched it on YouTube, that for genome editing what you need to know is the target site that you want to edit. So that means you have to have the genome sequence, the gene precisely which you want to edit. This information must be available for doing this exercise. So those crops where we don't have this information, the information has to be generated. Also, there is a general belief that the genome editing can be an alternative to transgenics. So there again you have to see, like genome editing is a tool which is much more precise, but it can edit the genes which are present in the plant. Okay. Suppose if you wanted to create a genome edited product for a gene like Bt gene, the Bt is not there in plant, it comes from a bacteria. So you cannot do editing for Bt. Now if you have to do editing for Bt using genome editing tools, then of course you can take Bt gene, you can deliver to a targeted site, but that is as good as doing transgenics. So it is not different from transgenics, and that's where the three categories which I described as site directed nuclease 1, site directed nuclease 2 and site directed nuclease 3. The 2 and 3 are almost like transgenic approaches only, but the first one, SDN1, which some examples I will describe, and also the fact is that nature has been doing this genome editing for long. For every gene, a gene is the basic unit of heredity, and the alleles of genes are defined as the alternative form of a gene. Okay. Now, so every gene has an alternative form. Now, this alternative form is created by mutation. There is no other way to create a new gene, it has to happen through mutation. So, for all the genes that are occurring in nature, for example, in rice if you have 37,000 genes, there are number of alternative forms of each of these genes present in nature. Some we have explored, some we have not explored. So those which we have explored, we know that these are the alternative forms. For many we don't know, but they are present, and nature has done that editing to create the alternative form of gene or alleles. By genome editing also we are creating alternative form of a gene. Now many of these alternative forms which have been created by nature, they have been effectively utilized by the plant breeders in the breeding program. So I am going to cite you some of the examples where naturally genome edited product by nature have been utilized in the breeding program for product development, and what are the difficulties that we face there and how we can overcome those problems when we go for genome editing as a tool in breeding program. So it's not getting forwarded. This one? No, this is not. Okay, now it is forwarding, fine. So before that I will just give you some glimpse of what work we have been doing through molecular breeding. Our major work focuses in the area of improvement of Basmati rice, and you know that traditional varieties of Basmati rice, they are very tall, they are photoperiod sensitive, and they are prone to lodging. So this is a Basmati crop which has fallen flat on the ground, after the grain formation, if there is a wind, it will fall generally, and because the quality becomes very poor, and their yield is hardly 2 to 2.5 tons per hectare, very low yield. So these were the major problems of traditional Basmati, and that is where in late 60s, Dr. Swaminathan, when he was head division of genetics, he conceptualized the idea of combining the quality of traditional Basmati into high yielding background, and the crossing program was initiated to bring again the alternative forms of a gene, that is plant height gene SD1, An alternative form was a DG Eugen dwarfing gene that was also created by nature. Okay, it was available in variety DG Eugen gene was available in Taichung native 1 variety, and that was crossed with the number of other genotypes, and we also crossed with Basmati, and the dwarfing gene DG Eugen dwarfing allele was brought in Basmati. So Basmati became dwarf, it became non-lodging, it became highly productive. So the first variety in this series was released was Pusa Basmati 1 in 1989, 1121 in 2003, and improved Pusa Basmati 1, where we collaborated with National Research Center on Plant Biotechnology. Dr. Mahapatra was involved in this program, where two genes XA13 and XA21, they were incorporated through marker assisted breeding, and this variety improved Pusa Basmati 1 was released. So again here you see there is a allele of XA13. All the rice varieties they carry XA13 dominant allele, and they are susceptible. There are certain changes that make the gene recessive and confers resistance. Now some such varieties were identified. One such variety was Bhogjeera 1 BJ1, and from that the gene was identified, mapped and transferred. At that point of time people did not know what makes it resistant, but now precisely we know XA13 gene is cloned, we know the sequence where the change has taken place, and that change makes it resistant. So now if you can create that change by genome editing in any variety which carries the dominant allele of XA13, you can convert it into a resistant variety directly without going through marker assisted backcross breeding. Here we have to transfer the gene from a variety called IRBB55. Both XA13 XA21 genes were present there. Marker assisted backcross breeding was involved, and as you will see that what are the different steps and what are the problems. So this is what we did 1930, if you see 8 decades of journey, where the grain length on cooking was hardly 12 to 13 millimeter in the traditional varieties like Basmati 370. This is one of the traditional variety. But now in 1509 this grain length on cooking is almost 1 inch, 20 millimeter plus. So that gives high volume expansion. You can cook a small quantity of rice, you can serve many plates, and therefore hotel industry find it's very much beneficial, and that's how these varieties have spread over a large area. The duration through breeding was reduced from 160 days in Tarawadi Basmati to almost 120 days in Pusa Basmati 1509, and productivity was improved from 2 tons per hectare to 6 tons per hectare. Now this seizure-like graph in plant breeding is hard to get. Generally when we increase the duration, we increase the yield, but decreasing duration and increasing yield has been a challenge, what we call as an unholy alliance, or it's a negative linkage that has been broken, and we have been able to breed varieties with short duration and high yield, which has directly impacted the foreign exchange earning through export of Basmati rice. And currently India is earning about 32,805 crores of foreign exchange annually, and the total export of Basmati rice is about 5 million tons. But the challenges that we have in the breeding program are a large number of diseases like bacterial blight, blast, insects like brown plant hopper, sheath blight, bacane, brown spot, false smut, and name a disease, it is there in rice. Now the total number has gone to almost 18, 19 different diseases. 20 years back these many diseases were not there, but there is always a continuous race between the host and the pathogen, and the new races evolve, new diseases resolve, and they keep on developing. So these are the major problems, and to solve these problems we have effectively utilized the molecular breeding tool, what we call as marker assisted backcross breeding. Now backcross breeding is an approach of breeding where you have a very good variety, but it has certain weaknesses. It is lacking 1 or 2 characters. So that trait you want to bring from another variety, which we call as donor parent. And the variety which has all good characters but one is called recurrent parent. So this is donor parent, this is recurrent parent. You make a cross and then you back cross with the recurrent parent to recover the genome of recurrent parent, because you want all genes from recurrent parent but one gene from donor parent to rectify the defect of the recurrent parent. So you will have to go for 2-3 back crosses depending upon how much is the diversity between the donor and recurrent parent. If donor and recurrent parent are too diverse, the number of back crosses required will be many. If they are very close, then few back crosses will be required. The steps which are involved in this process is, one is called foreground selection, where we select for the gene of interest that we are transferring from donor parent. The second is recombinant selection. So recombinant selection is important, and these are the problems which can also be addressed by genome editing. So for example, if we are bringing a gene donor parent from here into recurrent parent, this gene will come with the flanking genomic region from donor parent. From donor parent we want only one gene, which is a good gene. We don't want rest of the flanking genomic region, but the transfer of that segment will happen only through homologous recombination. So depending upon how close or how far the two recombination events are taking place, you may have a large segment of the donor along with the gene, you may have a small segment of donor along with the gene, but you want to have a minimal donor segment and the gene of interest. Okay. Now bringing this donor segment creates a lot of problem, because if there are undesirable gene in that region, then the performance of your recurrent parent goes down. You are not able to recover all the characteristics of recurrent parent. So that becomes a problem in the classical approach. Even if a naturally created favorable allele by spontaneous genome editing is available, while you transfer through backcross breeding, you bring lot of linkage drag, and that adversely affects the performance of recurrent parent. So this is one of the major problem in using, even if a naturally occurring gene is available, still you like to go for genome editing, because in genome editing you are selectively altering that particular gene and you are not touching anything else. So then we call recombinant selection. That means this is done to reduce the flanking genomic region. Then we have the background selection, where we try to recover the genome of recurrent parent to the extent possible, and generally recovery should be more than 95%. And at the same time you will have to practice the phenotypic selections for target trait, for yield, quality and stress resistance. All these things have to be done. So this is a regular typical marker assisted backcross breeding program, which involves these four key steps in transferring a gene from donor to recurrent parent. So that's what we did in case of improved Pusa Basmati 1 development. In Pusa Basmati 1, which had become highly susceptible to bacterial blight, the two gene XA13 and XA21 were transferred. Now if you see here, what we are talking about linkage drag, this is gene XA13 here. This is one isogenic line. This is chromosome 8 on which XA13 gene is located. This is XA13. Now if you look at the donor segment here, in this particular isogenic line, the donor segment is much bigger, which is represented by light color. In this case it is smaller. So if you have to select between these two lines by doing this background analysis, then you will select this line, because this also has XA13, and along with XA13 the donor segment is much reduced here. So that is how this analysis helps in identifying a plant with minimal donor segment plus gene of interest. So likewise we transferred these XA13, XA21 gene in Pusa Basmati 1121, which also had become susceptible to bacterial blight, and recovered all its quality. So this is original 1121, this is the donor that we used for XA13, XA21, and the new line which has been released for commercial cultivation in the name Pusa Basmati 1718. So this is an isogenic line of 1121 carrying two genes for resistance to bacterial blight. So in any line, if you create a genome edited line, the genome edited line is also in true sense an isogenic line. The best form of isogenic line can only be created by genome editing, where you don't do any other alteration except the gene of interest. In these alterations we are still getting some donor segment. So likewise another variety 1728, which is the improved version of Pusa Basmati 6, where we also incorporated XA13, XA21, and this has also been released for commercial cultivation, doing very well with the farmers. And you will find, if you just Google Basmati 1718, you will find thousands of videos. These are videos for 5 minutes, 6 minutes, which have been produced by different entrepreneurs. These could be people like you, farmers, traders, and they are available, and each of these have got a viewership of 50,000, 60,000, 1 lakh and so on. So that helps in popularizing the varieties a lot. One aspect which we have not really known so far, that how much is going in the background and what are the different stakeholders, and therefore we need to What is important, why we need to tie up with these stakeholders who are doing this job, is that when they are providing information about the varieties, because many times when I watch these videos, I find that they are communicating some wrong information. They are telling the information which is not correct about the varieties. So those things you need to tell them that is this information, you are doing good job, great job, but the information must be correct, and therefore we need to follow up with them. A new gene for bacterial blight resistance we have identified in a germplasm line called BAM72. This is bioprospecting of genes and allele mining. We had a project. So this germplasm was resistant to bacterial leaf blight, and on inheritance analysis it segregated 3:1 ratio, 3 susceptible to 1 resistance. That means it was controlled by a recessive gene. We followed QTL-seq analysis, and from QTL-seq we mapped this gene, and there are these possible 4 candidate genes that we have identified. Now we are looking at the expression profile to identify a novel alleles. This is conferring resistance to those races of bacterial blight which knock down XA13 and also XA21, and therefore having additional allelic diversity is very, very important. Now many of these alleles which nature has created, they are there sitting. They will not be known unless and until you challenge them with the pathogen. Otherwise, they will remain silent. So that's where the screening of the germplasm is extremely important for many of these biotic abiotic stresses. Another major problem is blast disease. So blast comes as a leaf blast, it comes as nodal blast, it comes as neck blast. And what picture you see here is neck blast, where all the the pancreas, they have broken from the neck and they are just drooping down. And because of this, now all the grains will dry up, hardly there will be any production in these varieties. The farmers, to prevent this disease blast, they use a fungicide called tricyclazole, and tricyclazole there is a strict limit of residue. In European Union, they are not allowing, if basmati rice has got tricyclazole 0.01 ppm. So 1 milligram of tricyclazole in 100 kilogram of rice, if it is present, they don't accept the basmati rice from India. Now if that is the kind of stringency, the only way you can tackle these problems of pesticide residue and rejection of basmati consignment is by genetic resistance, because once you create genetic resistance, then there is no need to spray fungicide, and if you don't spray fungicide, the residue problem will be solved. So that is where there are a number of naturally occurring genes that are there: PI1, PI54, PETA, PI5, PI9, PI2, PIB. They confer resistance to blast diseases, right? Now they have been identified, the donors have been identified for these genes, and through back cross breeding, again we have transferred this to one basmati variety. This is Pusa Basmati 1, 3 genes in Pusa Basmati 1 background from this donor, PI5, PI9, PI2 and PIB, and ultimately now we have got Pusa Basmati 1 with 1 gene, 2 gene, 3 gene, 4 gene, up to 7 gene pyramids in the background of Pusa Basmati 1. And depending upon the necessity, you can go for deployment. If one is broken down, then you can go for deploying the other gene. Not necessarily you challenge all genes to pathogen at the same time. So this can be a stepwise strategy to deploy them for the purpose of resistance. I was telling you about the bad characters from donor. Now you see here is a donor for PI9. This comes from Oryza minuta, a wild species. Now this donor has got red pericarp. It has got chalky grains. On cooking, grains will swell. So when you use such donor for transferring a gene, the entire quality characteristics of basmati is spoiled. Then to recover that quality character back again, in these are the lines which have got resistance along with the quality of basmati. That is where extensive background selection, phenotypic selection is very much essential. And you can get rid of all these problems if you are able to create these novel alleles by genome editing, because these genes are all rice genes. They are present there, except that you have a susceptible allele of that gene present, and if you can create a resistant allele by editing, then you need not to face these problems. So after you can see that this is Pusa Basmati 1 in Milan. This is a uniform blast nursery. All plants are killed because of blast infection, but when same variety is transferred with PI9 gene, nothing happens. They are all highly resistant. So this variety, Pusa Basmati 1637, we released for commercial cultivation, and the average yield of this variety is about 28 quintals per acre. The farmers are quite happy, and this you don't need to spray fungicide on this. Again, DG visiting the farmers field where we had grown in 2017, very bumper crop of this crop. Now, coming back to the background selection and the residual heterozygosity or residual genomics contribution that remains from donor parent, depending upon what kind of marker system you are employing to assess the recurrent parent genome recovery. Now, here you can see, this is chromosome 1, a isogenic line which is analyzed by SSR marker, pair of isogenic lines, and the same line analyzed by a SNP marker where we have used 50K chip developed by Dr. N.K. Singh. Now, if you use just a few markers, about 60 markers, microsatellite markers for background recovery, you find that this chromosome looks completely clean and you get an impression that there is no donor segment present here. But if you increase the number of marker to 50K, 50,000, then you find that almost this line is having so many donor segment present here, and almost all chromosomes you will find that there is lot of donor segment present. And this is so because if you take even 95% recovery, the 5% of 389 million base pair is a huge genomic contribution. So that will always be present, except that most of this is not phenotypically expressed, and therefore it is present there but you don't know. But through molecular tool you can identify where is the alleles coming from the donor segment is still present and how much efforts you need to put in to clean these. Do we really need to do it, or it is not necessary to clean? Unless and until it has got a adverse phenotypic effect, there is no need to invest too much of time on cleaning this. But if it has got an obvious adverse effect, then of course we have to do that cleaning exercise. Another disease is Bakane. It is caused by a fungus called Gibberella fusicori, which produces gibberellic acid in the plants, and because of gibberellic acid production, these plants grow tall and finally they dry. So you can see this is a farmer's field, and you can see this fields look so ugly because of the plants infected by Bakane. They have grown tall and they dry it. So we initiated a program on this. We identified a donor 1342, which again carries a naturally edited allele, and through mapping using a recombinant end-bed lines, we were able to identify 3 major QTLs, and one of these was QBK 1.2, which controls about 24% of phenotypic variation, which we have now transferred to Basmati background. Another area where we are working on is on what is called direct seeded rice. You know that rice takes about 3,000 liters of water to produce 1 kilogram of rice, and often rice is accused for being a water guzzling crops. Now, one of the potential alternatives is to go for dry direct seeded rice, so that the amount of water required for transplanting can be done away with. But when we go for DSR, one of the major problems that we face is the infestation by weeds. So weed is a major problem, and therefore we have been working in the area of developing herbicide tolerant rice. Dr. Reddy is also working in this area through genome editing and through transgenic route. But here we resorted to mutagenesis, EMS induced mutagenesis in a variety called Nagina 22, and a mutant this was a DBT funded project, network project, number of centers participated. Dr. Robin from Tamil Nadu Agriculture University was partner. Unfortunately, we have lost him, and he was able to identify a mutant plant. Here you see we had almost grown in 5 acres after giving the treatment with EMS, and then M2 seed was raised in the 5 acres area, and the whole plot was sprayed with herbicide imidazolam. And after spraying that, some 2 plants were identified which were resistant, and these plants were then potted, and finally we did mapping, characterization of gene, and we identified at 623 positions there is amino acid change that makes it resistant to herbicide in the gene ALS, acetolactate synthase. So now if we know the acetolactate synthase gene, its complete sequence, and we also know that if we alter certain positions in this, we create a herbicide tolerant version. Why it happens? Because the ALS has got activity for synthesis of 3 amino acids. These are branch chain amino acid, leucine, isoleucine, and valine. And the ALS molecule also has a herbicide binding site. So if you in normal rice varieties, herbicide binding site is active, and therefore when you spray imidazolpyr group of herbicides, the herbicide binds with ALS. As a result of allosteric inhibition, the ALS is not able to synthesize leucine, isoleucine and valine, and therefore these plants die. But in mutagenesis process, the ALS herbicide binding site is altered, and now if you spray herbicide, the herbicide does not recognize ALS, and therefore ALS is able to synthesize leucine, isoleucine, valine, and these plants survive without any problem. So that's what we did, and then after that we transferred this gene through backcross breeding. It was identified in the variety called Nagina 22. It's the upland variety, very bold grain. It's a drought tolerant variety, but quality is very poor. So from there we transferred to Basmati rice varieties, Pusa Basmati 1121 and Pusa Basmati 1509. So you can see here, this is a plot which is unweeded plot where no spray is done. It is full of weeds. This is original 1121, and this is 1121 carrying mutant allele of acetolactate synthase. So after spraying herbicide 18 days after direct seeding, these 1121 is completely killed. 1121 carrying mutant allele of AHAS is surviving. Likewise in 1509 also this gene has been transferred, which survives without any death. Here weeds are also killed, and also the rice plants are also killed. So now with herbicide tolerant rice that we have developed, it becomes possible to go for direct seeded rice in Basmati with a very effective weed management, and therefore no productivity loss. So now these lines have got excellent grain and cooking quality like 1121 and yield on par and with resistance to herbicide. Likewise in 1509 we have transferred that gene, and you can see here 1509 original is completely killed, but those carrying the ALS mutant allele, they are highly resistant to herbicide, nothing happens to them. Now the major challenges that we have, as you know, as I said, that these are the major problems: Bacane, sheath blight, brown spot, false smut, neck blast, brown plant hopper, yellow stem borer, leaf folder, root knot nematode. Among abiotic stresses we have drought, salinity, heat and nutrient use efficiency. And almost all these traits where the genes are well characterized, genes are known, genomic sequences are known, gene sequences are known, location of gene is known, they can be targeted through genome editing without getting into the all the problems of conventional plant breeding. Even if we are using molecular assisted backcross breeding, you have to spend a lot of time and efforts. I think I can skip this. Okay. Now let's come to some of the examples for which the practical demonstrations will be given by Dr. Reddy, at least some of them. You can see here, for example, there is a gene called cytokinin oxidase, okay, and this regulates rice grain production. Now this is a variety called Koshi Hikari, this is another variety Hawataki, and another variety 5150. And you can see there is a clear-cut difference in terms of grain number in these varieties. Koshi Hikari has got less grain number, Hawataki has got high grain number, and 5150 has got a still higher grain number. If you see as compared to Koshi Hikari, the changes that have taken place in the first exon of Hawataki is a 6 base pair deletion here and another 14 base pair deletion. Now if this deletion takes place in the first exon, you get this kind of phenotypic alteration. Why it happens? Because this locus is responsible for cytokinin oxidase. Now if you downregulate cytokinin oxidase, then cytokinin level will go up, and cytokinin level if goes up, then you will have more branching and more density of grains production. Now so this is one allele which is created by mutation, and this is a natural mutation the nature has created. Likewise in case of 5150, and looking at the phenotype, you can tell that 5150 phenotype is better than Hawataki. So this also carries a allele of the same gene, but in this case the alteration is here in the first, second and third exon, where you have about 110 base pair, 11 base pair deletion. Okay. Now this creates an alternative allele. So within a gene you are creating two alternative allele. One allele is specific to Hawataki, other allele is specific to 5150. and both these have got enhanced phenotypic values. Now, this is a kind of post-mortem. Nature has done this job. We have done gene sequencing, we have done, we means whomsoever has published the paper, and they identified that this alteration is associated with this phenotypic change. They took this alternative allele, and by genetic transformation of Koshi-Hikari, they demonstrated that there is a corresponding increase in the grain number. Now, if we can create this change, a variety which has got low grain number, suppose it is having 20% less grain number, and in that variety you also know the sequence of that variety, that it carries the wild type allele of cytokinin oxidase, this locus, and if you can alter that allele by genome editing, you can increase the grain number in that variety. So, generally when we are doing varietal improvement, we are working at a margin of 5%, 10% superiority, but if you can directly have an increase in grain number of 20%, 25%, that will be a huge increase by altering a single gene. So, this is one case which has been successfully demonstrated by many people through genome editing now, and the examples are available, Dr. Reddy will tell you. Likewise, there is another gene called DEP1, dense and erect panicule. So, this is natural variation at DEP1 locus enhances grain yield in rice, and you can see here this is a near isogenic line carrying the recessive allele of DEP1. This is dominant allele of DEP1. So, the DEP1 dominant allele, it gives less grain number, and the altered allele, that is recessive one, it is giving high grain number. How high grain number is obtained in this? These are analyzed from a close angle. The internode length in the panicule is reduced, the number of branches are increased. So, you get more number of branches and the short internodes in racilla, and therefore, you get high grain number. And this happens because of 625 base pair deletion here, and this deletion leads to truncation of this protein. So, now you know precisely that if you are able to make this change, okay, then you are able to bring this phenotype. Not necessarily only this change can give rise to this phenotype, there could be some other change which also has the similar effect in terms of termination of the truncation of the protein, can also give rise to a similar phenotype or even better phenotype. So, in none of the genome edited product which I have come to know so far, that people have been able to create the original mutation that nature has created. Say for example, DEP1 has been edited, but in DEP1 having the, or for example cytokinin oxidase has been edited, but having the same 6 base pair deletion and 11 base pair deletion with a positive phenotype has not been obtained. You find some other change which also give rise to the similar phenotype. Now, that is where there is a question I often ask to those who are working in genome editing, that in nature such changes would have happened in large number, but nature has retained certain alleles, but not others. Others alleles have been eliminated by selection pressure. So, there must be some reason for retaining this allele through the evolutionary process, and can we make it so specific, and that probably Dr. Reddy can answer, I cannot answer, that we make only specific change of 6 base pair deletion and 11 base pair deletion and no other change. So, that becomes in true sense a site directed mutagenesis. Still we are directing to the site of gene, but we are not able to direct the change that we want to introduce. So, that change process is still by and large a random process, and then you screen, and some of them happens to be a desirable phenotype. So, that specificity has to be worked out. Likewise, another gene, that is OSPL14, which is a site for microRNA 156, and by altering this gene here, if for example, if you have a T in place of this is the pink one is black is T and G. So, this change, if you have G and T, if you have change, then there is a loss of site of this microRNA, and therefore, there is differential regulation, and this again gives rise to large panicule size. Likewise, a gene called this DST, which controls drought and salt tolerance, and it has been created by genetic transformation, potentially demonstrated both the effect by this gene and which particular alteration leads to this change is also known. So this really gives you such a powerful tool to create n number of alleles of a given gene with positive phenotypes. It is for you to create those alleles, screen them for phenotypic values, and then identify the lines which has got the best phenotypic expression for utilization in the breeding program as a direct product or subsequently in the breeding program. Another such classical example is of a gene called GS3, grain size 3 genes, and this grain size G3 happens because of a change from C to A substitution 165 base pair downstream from the predicted translation start site. Okay. Now this C to A change makes grain very long. This also causes a premature termination of the peptides, polypeptides, and because of that the small grain is a normal wild type allele, and in that small grain type if you change C from A, then you get a long grain. So this is the lines where they have demonstrated. But when we analyzed this in our material, this is our material, this is a variety called Sonasal, and this is variety Pusa Basmati 1121. And you can see how potential is this one single nucleotide polymorphism from C to A that increases grain length in such a proportion. Grain length before cooking, grain length after cooking. So this has been very clearly demonstrated. In addition to this, we also analyzed the gene in these two varieties, and we found another polymorphism in 3' UTR region, but the contribution of 3' UTR polymorphism is much less. The maximum contribution comes from C to A polymorphism. So now if you want to make a short grain variety like Samba Masuri and want to convert, because Samba Masuri has got excellent grain and cooking quality, okay, people like its taste, but if you want to make it a long grain variety, you just change from C to A without altering any other character of Samba Masuri, you make it long grain. And if somebody wants to have a rice which is without aroma, but it feels like, looks like Basmati, you can create that kind of long grains. Those are the kinds of opportunities that are available. Likewise, I was referring to XA13. In XA13, these are the lines which carry XA13's dominant allele, and those lines, these are susceptible lines, and these are the lines which are highly resistant. And in this promoter region, there are the number shows the number of bases deleted or added depending upon the color of triangle, and these are the different alternative forms. Each variety is resistant, but each one is having a different polymorphism in the promoter region of XA13 gene. So you can create those many alternative alleles of XA13 and have resistance phenotype for bacterial blight. So you need not to go for backcross breeding. You just pick up a variety which is susceptible, for example Pusa Basmati 1. Earlier to make it resistant we had to transfer the gene from IRBB 55, a non-Basmati. Now we can just do genome editing of XA13 in Pusa Basmati 1, because Pusa Basmati 1 carries the dominant allele of XA13 which confers susceptibility, and alter it, make it resistant in one go. So those are the kinds of challenges which are available and would have a lot of practical values. So this work has been done, Dr. Reddy is doing, he will present his work, but also I was a member of a committee of a DVT sponsored project with Mahiko Research Foundation, and they have been doing the editing of the genes called DEP1, improved Pankill architecture 1, DST, GN1 and GS3. These genes in the one of the cytoplasmic male sterile line and its B line counterpart, fertile counterpart, that is 58025B. So in 58025B they have created genome edited lines, and you can see here generally the 58025B has less number of grains, and here the grain number is increase is substantial. Almost it close to double. The effect of the allele would also vary from genetic background to genetic background. So on an average 25B will have about 125 to 150 grains, but these edited lines they have got a grain number 200 plus. So now by doing this it is possible to improve the productivity of the CMS lines, because if you can have the seed yield production ability enhanced, then the cost of hybrid seed will go down. So this has been demonstrated in this case, in case of 58025B. Likewise, I said herbicide tolerance. So here also we understand now what change will make it herbicide tolerance, and selectively that change can be obtained and you can directly create the improved version. Now let us see, when you do this exercise, what are the different possibilities that are likely to happen. So for example, this is say chromosome 1, where you have the target site which is to be altered. Let's take, I have taken this example for C to A change for grain number gene, okay, sorry, for grain size gene, okay. So this original wild type is C. Now suppose this is present on chromosome, just a hypothetical example I have taken, it is present on chromosome 1, and you do a CRISPR-Cas9 transgenic lines you develop, your Cas9 integration will happen on some other chromosome, because that insertion process is by and large random. You are not able to direct that you want it to be inserted at a given position. So like any other transgenic events that you do, it will get inserted on chromosome 9, say chromosome 5 for example. Now after that you develop a T0 plants. The T0 plants will produce seed. The seed harvested from T0 plants will be T1 seeds. Now what are the possibilities in terms of the genetic constitution of these seeds at two loci? You are dealing, one locus is the target locus that you want to alter, and the other locus is where the insertion of Cas9 has taken place, because ultimately you have to get rid of CRISPR-Cas9 by segregation analysis. So for example, CRISPR-Cas9 edit lines will have at GS3 locus, they can be CC, that there is no change taking place. That is one possibility of so many transgenic lines that you develop. One case, they can be heterozygotes because there are two homologs, and it is highly unlikely that you will be able to bring a precise change at both the homologs at the same position. That is unlikely to take place. So this situation will not happen. Most likely, if it happens, change happens, it will happen like this. And with Cas9, it can be, because this is coming after one generation of selfing, so Cas9 locus can be in homozygous condition, it can be in hemizygous condition, and it can be in null condition. Ultimately your goal is to, so you will have three possibilities here, two possibilities here, two possibilities here. So total combinations are 6, different genotypic combinations are possible. Theoretically there will be 9 combinations possible. Ultimately you are interested in identifying a plant which is null with respect to Cas9 locus, that is eliminated by segregation, and it has altered homozygous allele A in the this locus on chromosome 1. So this is the plant that is of your interest, and you have to correlate its phenotype in terms of grain size, how much change has happened, and there will be number of phenotypic alterations that will happen, and this itself will be many types. Some may be having a, if we can do that C to A alteration, but the same phenotypic changes can happen because of other alteration also, okay. So if we can be as precise as C to A, fair enough, then we get the natural allele created. If not, then we get some other alternative alleles created, but that allele also may have the exactly similar phenotype or even sometimes better phenotype. So this lot of screening has to be done to come out with the most viable product that you would like to take forward for direct releases or for utilization in the breeding program. So I think this is in brief what I wanted to cover and tell you what are the possibilities. See, for example, when we talk of transgenics versus genome editing, you have Bt cotton an example, but the Bt gene comes from the bacteria. So if you wanted to create SDN1 type genome editing product for Bt, it is not possible because Bt gene is not existing in the cotton plant. It has to come from outside. So there is no solution for that. So if general perception is given that the genome editing is alternative to transgenics in all cases, not so. The genes which are existing can only be edited, those which are not existing cannot be edited. They have to be brought in from outside, but possibly in much more targeted manner, okay. You can, if you can direct the site of integration, it will be itself a great achievement, because to create Cocker 312, one event which has been used by hundreds of companies in backcross breeding program, you know, Monsanto would have spent a lot of time in screening large number of events to come out with one elite events. Now, the genome editing can help you in creating elite event in a higher frequency if you can target the insertion sites in such a region where there is no shutting down of the native genes or there is no disruption of native gene. If you can direct through this tool the integration site of the transgenes, I don't know whether that will be possible, when it will be possible, because that those who are involved deeply into it will tell. So I think with that I would like to thank you very much for your patient hearing, and if you have some questions I will be happy to address.
My question is that by gene pyramiding, how much time it will take to create a new variety?
So generally, at least 2 back crosses you have to give, and minimum 3 years time with 2 crop season. At least 6 crop season will be required, at least.
Okay, so
and then testing another 2 to 3 years. So it will not be before 5 to 6 years, 5 years that you can have a product.
So gene pyramiding can be done for 2 different traits, or can only for single trait?
No, so pyramiding, when we say pyramiding, is for multiple genes. Okay, so you can have bacterial blight, you can have blast, you can have yield genes, you can bring in whatever you want. Basically, as long as the genes are defined, donors are known, and markers are known for that, you can bring them together. But the term pyramiding is used in a strict sense, and it is used in the context when you use the genes which confers resistance to different races. When you bring them together, it is called pyramid. Otherwise, in loose term, we call them stacking or multiple gene this thing. But the term pyramiding is when you have multiple races and the genes conferring resistance to each race individually, and you bring them together. That's what is called pyramiding.
So here we also perform that marker-assisted back crossing, that recombinant selection, foreground selection in case of gene pyramiding.
Yes, yes.
Okay.
So it depends upon, if you want to pyramid the genes in a very good known variety already, which is lacking the genes, then you have to go for back cross breeding. But if you want to develop a new product altogether, then you can make a biparental cross, and you need not to go for back crossing, and from that you can derive a altogether new variety, which will not be similar to your donor parent or recurrent parent. It will be a different variety, but having the gene of interest from the donor parent.
Thank you, sir.
Thank you, sir, for this nice lecture. My question is here in the grain size and branching of the panicle, where we have seen the DAP1 and cytokinin oxidase. Both are working in the similar way. So I was thinking that are they related, these 2 genes?
No, these are the DAP1 pathway is different, Dr. Reddy. I think that pathway is different. I have detailed there. This one that is not regulating cytokinin oxidase. That pathway is different. Now one would know, like when you bring these 2 together, DAP1 and cytokinin oxidase, then what kind of compensatory mechanism happens, that is something to be seen, and that will be very interesting. Not necessarily 1 plus 1 is 2 always, it can be 1 plus 1 can be 11, or it can be sometimes negative interactions will also happen. But only after bringing them together one would know what will be the effect. Yeah.
So cytokinin is regulating the meristem development. That is actually involved in meristem cell gene conjugation. Cytokinin is upstream regulator. It regulates many other processes also.
Thank you.
So for herbicide tolerant variety also, right? So that gene ALS2 is very specific for a broad spectrum of herbicides, or it's very specific to particular kind of herbicide?
This is effective against the herbicides which is called imidazolinones, which target the acetolactate synthase. Okay. And these are again broad spectrum herbicides. In terms of their weed efficacy control, they are very broad spectrum. But it is effective for this herbicide only, imidazolinone, which target amino acid synthesis pathway. There are different ways, these you can have fatty acid metabolism altered, you can have photosynthetic pathways, you can have amino acid synthesis. So this one is targeting amino acid synthesis.
Thank you, sir, for the wonderful talk, and it's always a new learning experience when we listen to you. I have, sir, two questions. One thing you told that by decreasing duration and a basmati yield has been increased. So what kind of duration has been decreased in this case, whether vegetative duration has been decreased or reproductive duration or both? And second question is, you told that you have developed herbicide tolerant rice for imidazole, acetolactate synthase, that kind of herbicide. And in previous slide you told that European Union and other countries, so they have banned trichlorozole containing tricyclozole containing that fungicide. So what do you think that in future they can ban if the herbicide contain these kind of basmati contain this herbicide?
Well, I mean, that's a general question with respect to the pesticides, whether it is herbicide, insecticide, or fungicide. And there are about 20 listed pesticide now in case of rice, which Basmati export is quite conscious of, and they are not permitting beyond certain. For every pesticide, you have got a maximum residue limit. As long as it is within that limit, there is no issue, but once it crosses that limit, then it will not be accepted. So now there are a number of other insecticide like buprofogin, which is used for brown plant hopper, is also listed in that. As far as herbicide is concerned, these imidazole group of herbicides, they are considered very safe because the ALS activity is not there in the mammalian system. So it is completely safe for the human system and mammalian system. Okay. So and then you have all these protocols before we go for CIBRC registration of new molecules. This herbicide is safe. It has been used in soybean for many years, more than 20 years. It has been extensively used in soybean, and absolutely no concern of safety there. So for every such molecule there is a concern, and then the protocol is to be followed for that. So, and you had some other question. First question was
Sir, that duration you told. That
so duration, it is like the overall duration from 160 days, it has been brought down to 130 days, and the onset of reproductive phase happens much early in these varieties. Yeah. So mostly it is because of controlling the photoperiod sensitivity. They have been made photoperiod insensitive, and they flower irrespective of when you plant them. Once the crop is 80 days, it will flower. While in case of the traditional basmati varieties, they are photoperiod sensitive. Irrespective of when you plant them, they will flower only when they get the short day length in the first week of October.
and sir, in glyphosate resistance transgenics, some genes has been transferred to weeds. So this herbicide tolerance rise. So these kind of gene can also go to related weed in future. So this ALS.
Dr. Reddy is going to talk about ESPS synthase in some detail. He will discuss about it. You see, that risk is always there, but that is where there is something called stewardship in commercialization of any product. Even, for example, delaying the development of resistance in bollworm for BT, you have a stewardship, you have refugia, and so many things. So likewise, when it comes to your concern is that from herbicide tolerant rice, if the pollen flow happens to a wild and weedy rice, the wild and weedy rice will also become resistant to herbicide. And that is where we have to be a little cautious in terms of deployment. Those areas where wild and weedy rices are existing, for example, if you go to northeastern states, you find the Oryza nivara, Oryza rufipogon, and the intermediates widely present in the field, in ponds, in the submerged areas. So there one has to avoid the deployment. But the areas which are completely free of, for example, you travel entire Haryana, Punjab, Western UP, you don't find any wild rice there. So there it is a safe deployment without any problem. And also there are other management strategies that you can bring in different routes of herbicide tolerance. For example, this is one. And along with that, if you can combine another mode of herbicide tolerance. So 1 year you spray the other herbicide, the next year you spray second herbicide. So if there is any resistance developed, happens in the weeds particularly. There is another problem is herbicide resistance develop in wheat, that can be taken care of.
Sir, my question in marker assistant backcross method, we go B5 and 4 and 5 generation. Why not select in F1 backcross first? Because in this at least one genotype have both the gene donor and recipient.
So in BC1 F1, you know, I often ask this question in my presentation, that there is a possibility, those who are from genetics would appreciate, the average recurrent parent genome recovery in BC1 F1 is 75% average, okay. But what is the range? Range is from 50% to 100%. So if you have a large population of BC1 F1 plants, large means you have 2,000, 3,000 plants, then there is a possibility to identify a plant which has the gene of interest, but the gene will always be in heterozygous condition. It will never be in homozygous condition, because one parent you are using is recurrent parent, other parent is donor. So only one is coming from donor, one allele is coming from recurrent parent. The plants which are positive for the gene of interest, they will be in heterozygous condition, but those plants can have recurrent parent genome recovery maybe 90%, 95%, are as good as recurrent parent. But then your population size has to be very, very large, number one. Number two, you have to do substantial marker analysis for all these plants to identify a plant which is very close to recurrent parent. If you are able to do that, that is the beauty of marker assisted backcross breeding, that you need not to go for 5 backcrosses. You can do with 2 backcrosses. Extreme cases you can do with 1 backcrosses, provided your population size is very large. But generally we don't deal with that large population size. The population size remains about 50 to 100 in backcross populations, and that is where we miss to get the such rare recombinants.
Thank you, sir.
Sir, I also have one question. It's regarding the grain size. In nature, in many genotypes, we have seen that the grain size is small. So, there is a advantage associated with that smaller grain size, if particular?
So grain size and grain number, there is a negative correlation. So when you have size increases, the grain number substantially decreases. Most of our Basmati varieties where size is big, for example 1121 has 95 grains per pencil, but if you take Samba Masuri, which has 250 grains per pencil. So it is a overall balance of source and sink, but if we have independent genetic control, okay, then there is a possibility to increase the grain number to certain extent. For example, this one QTL Dr. N.K. Singh has mapped 4.1. Now we have transferred 4.1 has been transferred to number of varieties, and there is a increase in grain number, but it does not happen independent of other grain quality characteristics. The moment we increase grain number, the grain becomes thicker also, the quality goes bad. So one has to really do that exercise and select the plants which are most desirable.
Thank you, sir.
It's a very complex interaction. It would not happen that easily actually.
Thank you very much, sir, for the excellent lecture. I think we can discuss later. After tea, we have to start another lecture. Sir, thank you very much. Thank you for sparing your valuable time and coming to us. So, just this certificate.
Thank you.
Thank you.